Hello, dear student. Myself Arif Khan Pathan, working as a senior lecturer, MC Society Institute of Pharmacy, Diploma Azam Campus, Camp Pune. So in this topic, we'll see the antibiotics. As far as the antibiotic is concerned, first we see the discovery of antibiotic. Penicillin was the first antibiotic discovered, and that was an accidental discovery of penicillin. That is by Alexander Fleming in 1929, when Sir Alexander Fleming observed that the bacterial culture contaminated with the molds inhibited the bacterial growth. When he had grown the bacteria, and there was say inhibition of the growth of bacteria, in this slide it got clear. Alexander Fleming grown the bacteria, but accidentally there was a grown of some other fungus, and these fungus or a mold as produces some other substance, metabolically produces some other substance, chemical substance, and that substance has inhibited the growth of bacteria in these immediate region. And then Sir Alexander Fleming shown the conclusion, or he considered that there are some other substance which is being produced by the molds and which has an ability to inhibit the growth of bacteria. So further he isolated that chemical, and that is known finally, that has been given a name, that is penicillin, and penicillin antibiotic, and that was the first antibiotic which has been produced by the fungus and inhibited the growth of these bacteria, means produced by the one types of microorganism and inhibit the growth of another type of microorganism. So that was the first antibiotic discovered, that is penicillin, and the mold then further, or this fungus then further known as Penicillium notatum. So as far as the definition of antibiotic is concerned, that there was an accidental discovery or discovery part of invention of the antibiotics. Now this is the definition. Antibiotics are the chemical substances derived from or metabolically produced by one type of living organism and which are capable of inhibiting the life processes of other microorganism, and in small concentration. It means what they required in small concentration, as well as they are effective in small concentration. A little bit concentration is sufficient to inhibit the growth, and that is important as far as the dose is concerned also. So it is a good characteristic. Next, it also includes the synthetic compounds which are a structural analog of naturally occurring antibiotics. It doesn't mean that only antibiotics are obtained from the microorganisms and they are only natural in origin. They can be synthesized in the laboratory also, and that we call a synthetic antibiotics. Next, this is the classification of antibiotics, and depending on the spectrum of activity, that is, and the range of activity of these antibiotics. So that first is a narrow spectrum antibiotics. Narrow spectrum antibiotics have high degree of specificity. It means what, their range is narrow, working range is narrow, and they are effective on selectively towards either gram positive or on the gram negative. They are selective, as well as narrow range or narrow spectrum, that we are calling antibiotics. Example, benzyl penicillin is there. Next, broad spectrum antibiotics. So broad spectrum antibiotics has a broad range of their effect, and these antibiotics which are effective on the large number of pathogens or large types of bacteria, not specific to the gram or negative, or not only inhibit the gram positive, they are inhibiting most of all types of bacteria, that the working range is wide or a broad. That's why the spectrum that we are calling a broad spectrum antibiotics. Example, streptomycin, chloramphenicol, tetracycline. These are the examples of antibiotics which are broad spectrum antibiotics. So this is the classification depending on the spectrum of antimycobacterial activity, either a narrow spectrum or broad spectrum. So this is the one of the criteria to classify the antibiotics. Next, depending on their biosynthesis, this is the classification depending upon the biosynthesis. Biosynthesis means what are the precursor required for their synthesis. Depending on that, depending on the type of precursor and depending on the method of biosynthesis, this is the second way to categorize or classify the antibiotics. So in that, first, Antibiotic derived from amino acid means the amino acid is a starting material or a precursor for the synthesis of these types of antibiotics, and that's why we are saying that these antibiotics derive from amino acids. Next, the example from in these next example is the when they are derived from the single amino acid, chloramphenicol is an example. From two amino acid, benzyl penicillin, it is an example. The antibiotic derived from many amino acid, that is a polypeptide. Definitely, many amino acid will join together by the peptide bond. It means it will be a proteinous in nature or peptide in nature. That's why we are calling polypeptide types of antibiotics. Bacitracin, it is an example. Next, antibiotic derived from sugar. So basically the starting material is sugar. These antibiotics actually made up of sugar. So these are the amino sugar antibiotics, that is aminoglycoside antibiotics, streptomycin, neomycin, gentamicin. These are the antibiotics example which has a sugar and basically which is derived from sugar part also. So next, antibiotic derived from the acetate and propionate unit, that we call a tetracycline and erythromycin. When you see their structure, structure is complicated, even though we can say that when we see their structure, one can say that these are made up of multiple acetate and propionate units. Next, that is the last one, miscellaneous, griseofulvin and rifampicin. These are miscellaneous, where which there is no specific precursor for the biosynthesis of these types of antibiotics. So this is the second criteria to classify the antibiotic depending on the biosynthesis. Now this is the most important third criteria on which basis the antibiotics are classified, that is on the basis of its chemical structure, or one can say that this is a chemical classification of antibiotic. So in that first major class, that is beta lactam antibiotics. Now beta lactam antibiotics, on which basis we are calling beta lactam? Because they contains the beta lactam ring. Beta lactam ring is a four-membered ring which has a nitrogen as a heteroatom and oxo group. This four-membered ring we had seen in the heterocyclic ring also, and this is the beta lactam ring. Now these all antibiotic which has this structure or heterocyclic ring in its structure, that is called as beta lactam antibiotics. We have the example benzyl penicillin, then benzathine penicillin, phenoxymethyl penicillin, ampicillin, carbenicillin, cloxacillin, cephaloridin, cephalaxine, and cephalothene. So amongst these, one can say that till this cloxacillin, these all are penicillin types of antibiotics, whereas cephaloridin, cephalaxine are cephalosporin types of antibiotics. Now what are penicillin, what are cephalosporins? It will get further, it will get clear in further slides, but it will one can say that there is it's a relationship, or that there is a say differentiation, that penicillin types of antibiotics and cephalosporin types of antibiotics. Now this is the second class in the chemical class. First one was the beta lactam antibiotic, which has a beta lactam ring in its structure, whereas this second major class, that is the non beta lactam or non lactam antibiotics. Why we are calling non beta lactam? Because this doesn't contain a beta lactam ring in its structure, and when it has a beta lactam ring, we are going to categorize as beta lactam antibiotics. If it doesn't have, non beta lactam antibiotics. So definitely, if it doesn't have a beta lactam, then certain other heterocyclic rings will be present, and depending on that, they are given their subclasses, or the name of the antibiotic will be depending on the heterocyclic ring present in that. Obviously, it will be not a beta lactam ring, but it will be other than beta lactam. So whichever, like for example, antibiotic containing hydronaphthacin ring in its structure. So we have example tetracycline. Now this is the say naphthacin ring, hydronaphthacin ring, the saturated one again. So hydronaphthacin. So this tetracycline will have this heterocyclic ring in its structure. Means obviously it doesn't have a beta lactam. It has other ring, that is hydronaphthacin containing. Next, aminoglycoside antibiotic is there. That is this one, aminoglycoside antibiotics, streptomycin, neomycin, gentamicin. Chemically, they are glycosidic linkage they have. Next, macrolide. Macro means a larger one, larger complicated structure, that is erythromycin, is belong to the class macrolide antibiotics. Next, N-acetylmycin, rifampicin is one of the example which showing the presence of, or coming under the N-acetylmycin types of antibiotics. Rifamycin or rifampicin, one and the same. Next, polyene or macrolide antibiotics. Poly means many, ENE stand for double bond, so this containing a, and macrolide stands for complicated structure or complex structure. So one can say that it has a number of double bonds and the complicated structure. Antibiotics, that is called as poly and antifungal antibiotics. Also we have example nystatin, hamicin, amphotericin B. These are the example. Next, anthracycline antibiotics will have anthracene ring in its structure, like two benzene fused together, that is called as naphthacene, three benzene in same plane, that is called as anthracene. So these types of antibiotics will have an anthracene ring in its structure. Example, actinomycin D, daunorubicin, or adriamycin. So these are the example which are anthracycline ring containing antibiotics. Next, peptide antibiotics. Peptide means which has a polypeptide ring, means number of amino acids joined together. As we had seen in the biosynthesis, number of made up of number of amino acids, means they have proteinous in nature. We have called it as the peptide types of antibiotics. So this is the one of the class peptide types chemically, which doesn't have a particular heterocyclic ring, but they are having peptide linkage. That's why peptide types of antibiotics. Bacitracin, it is one of the popular example. Next and last, that is steroidal antibiotics. Steroidal antibiotics, fusidic acid is the example, which has a steroidal nucleus. One can see the steroidal nucleus, which is four membered. In this figure one can see that four ring fused together to form basic steroidal nucleus. So this steroidal nucleus means that will have steroidal nucleus in its structure, will not have a beta lactam ring. That's why we had categorized under the non beta lactam types of antibiotics, and particularly which it has a steroidal nucleus it has. That's why steroidal antibiotics. Next, nucleoside antibiotics. Puromycin is the example. Next, other heterocyclic ring containing antibiotics, mitomycin, cycloserine, which has not particular heterocyclic ring or specified. It is not specified. Then we are calling other heterocyclic. Non-classifiable, griseofulvin and chloramphenicol. In such a way, this was a chemical classification of antibiotics, that is beta lactam and non-beta lactam. Next, the mechanism of action of the antibiotics, particularly beta lactam or penicillins, how they are acting. We have to see the animation also. The basic thing is that beta lactam antibiotics work in inhibiting the formation of peptidoglycan cross link in the bacterial cell wall, and hence they are weaken the cell wall and bacterium of the bacterium, and in other words they are causing the cytolysis, means breakdown of the cell, and due to that there is a growth of osmotic, due to the death of that causes the death of bacteria due to the osmotic pressure. Osmotic pressure it get increased, and there is a breakdown of the beta lactam ring. The animation, specific animation we have to see, and that is already given the link. Next, this is the general structure of the penicillin types of antibiotics. In the next thing you can see that next image, it is made up of the penam ring. So penam ring consists of the thiazolidine and the beta lactam. Red highlighted ring indicate it's a beta lactam, and this was a 5-membered ring in which the common nitrogen, sulfur and nitrogen is there, and which is a common one, saturated. That's why thiazolidine ring and the beta lactam ring fused together. One can see that this is the penam ring. Penam is a heterocyclic ring. Actually we had seen in the heterocyclic rings also, and this penam ring will be present in all penicillin types of antibiotics. So basically it will be a beta lactam antibiotics, because beta lactam ring will be there, and plus these thiazolidine, it indicate that the fused ring structure, that indicate it's a penam, and it is made up of beta lactam ring, which is a 4-membered ring, and 1,3-thiazolidine or thiazolidine ring, actually thiazolidine. So they both will 5-membered and 4-membered ring fused together to form this fused ring structure, that is called as penam. And most of these antibiotics will give the penam, will show the presence of this penam ring, and penam containing beta lactam antibiotics are called as penicillin types of antibiotics. So this is the structure of penicillin. So just we had seen that it has a penam ring. In addition to that penam ring, penam heterocycle, it has other groups also, which is like a somewhat common one. So we'll give the numbering. We'll start a numbering as far as the penam is concerned. So obviously it will start with sulfur 1, 2, 3, 4, 5, 6, 7. Now the substituents are concerned. So at second position there will be two methyl group. At third position COOA, that is carboxylic acid group, carboxy group. And at sixth position there is amino and acyl side chain that we call amino bridge, amino acyl. RCO, it stands for acyl. So amino acyl side chain at sixth position. At third position COH group, and at second position there will be a 2-methyl group. So these are the common components of all penicillin types of antibiotics. Common structure and numbering is as per the rule, as per the numbering rule. Now this penicillin beta lactam ring, just we had discussed that penicillin contains a beta lactam ring which is a 4-membered ring, and thiazolidine ring which is a 5-membered ring, and the nitrogen is a common adjacent carbon, and both the rings are common actually in the bicyclic ring, and that we call a penam. Now it's a fused ring structure which is made up of beta lactam and thiazolidine, just we had discussed. So most of points we have discussed actually. Next, it's like for example, both hydrogen at C5 and C are alpha orientation and they are folded. This is most important. The ring is folded at C5 and N4 axis, means at the common side the ring is folded and it looks like this. One can say that this is say 4-membered beta lactam ring, and this is the 5-membered thiazolidine ring. So when they are fused together at the common points, there is a say folded structure, and this structure of common structure of, we are just talking about common structure of beta lactam is looking like a flying bird. It looks like a flying bird, you can compare it, okay, but one can remember that it looks like a flying bird, and it has all components of beta lactam ring. Just we had discussed acyl side chain and this. Now the common term is coming that 6 amino penicillinic acid, 6 amino penicillinic acid, this completely with the beta penam ring which has 2 methyl group, and the carboxylic acid group, that's why penam with carboxylic acid group is called as penicillinic acid, and which has on the sixth position there is a say amino, that's why 6 amino penicillinic acid. And the COR, the acyl side chain, we are not taking into the consideration by this part. So we are calling that this is the beta lactam ring, this is thiazolidine ring, and this is the acyl side chain. So this is a common part in all types of penicillin types of antibiotics. So next part we'll see that how these all penicillin types of antibiotics can be derived from this common structure, and what are the changes, what will be the role of this R or alkyl group, which will be the alkyl group, and what will be the then antibiotic is forming. That we will discuss in the next part. Okay, thank you so much.